Okay, someone
Yes,
has been. Yes, thank you. Okay. Thank you so much. So, my first question will be: is there anyone that can differentiate these following words to me? What is DNA? What is chromosomes? What is nucleotide? What is nucleoside? And what is nucleic acid? Anyone?
Yeah, I can try.
Okay.
So you add DNA, so we know this is basically what carries information from cell to cell. It is made up of chromosomes. Yeah, I think so. Or chromatids, one of them,
Okay.
called
DNA.
around and condensed.
Okay.
Yeah, so that gives you DNA. So in your chromatids, yeah, they are made up of nucleotides. Yeah.
Okay. Go on. You're actually on the right path. Yeah, sorry.
So
Go on.
yeah. Your chromatid made up of nucleotides, and then I think you asked what else did you ask?
Nucleotide, nucleoside, and nucleic acid.
Yeah, the nucleotide and nucleic acid now. But then from the DNA up to the nucleotide, at least I know what goes on.
Okay. Any other opinion? Does anyone else have a different opinion from what was said by your name, please? Joshua, right? The person that just finished. Okay, Joshua. Thank you. So any other person has a different opinion? Please talk so I don't worry. There's no right or wrong answer. You can always tell your opinion. Don't be shy. So, okay. Basically, this is how I'm going to put it. So this is how I'm going to put it. So I'm very sure you guys are wondering what those diagram means. The first one on this line, this is a DNA. This is a chromosome. Oh, sorry. Yeah, this is a chromosome, and also this is actually a chromosome. But I'm going to explain the difference. A DNA is like the genetic material of the cell that is in the nucleus, right? That is a DNA. So when two DNAs compress together with some structures that is known as chromatin. So this is a DNA, like one single linear like this. This is a single strand of DNA. So when two strands of DNA, that is a double strand of DNA, wraps around histones, these histones then compress this DNA into a very smaller structure. That is what is known as chromosome. Do you guys understand? So this, I intentionally put this building block. These building blocks that make up the DNA is what is known as nucleic acid. So think of it, it's how do I say it? It's like a building block. The nucleic acid comes together to make the DNA. Then the DNA, two strands of the DNA wrapped around an histones come together to make a chromosome. Now, that will take us back to today's topic, which is nucleic acid. And I asked the first question, which was the difference between nucleotide, nucleoside, and nucleic acid, right? So I'm going to start by sharing my slide now so that we can go on. I'm sorry, I'm a little bit. Can you guys see my screen? Can you see my screen, please?
Yeah.
Yes.
Okay, thank you. So basically, a nucleic acid are like the basic units of a DNA. They come together to make a DNA. But the nucleic acid itself has some component. That is, the nucleic acid is what we call DNA or RNA. Do you understand? So a DNA or an RNA is like a group of nucleic acids coming together to make a DNA. But the nucleic acid itself is made up of nucleotide. So nucleotide are like nucleotide are building blocks for nucleic acids, and the nucleotide also has its own building blocks. That is, sorry. Oh, sorry about that interference. Please, let's continue. So, the nucleotide, a nucleotide is a building block of three components, and the three components is it's a sugar, a nitrogenous base, and a phosphate group. That is a nucleotide. A nucleotide is made up of a sugar. This is the sugar. I can't write on this thing. Sorry. The nucleotide is made up of a sugar, a base, and a phosphate group. The difference between a nucleotide and a nucleoside is the addition of the phosphate group that you guys have heard. Okay. I'm sorry, I'm trying to. Sorry. This thing is annoying. This thing. Sorry, I'm just going to use my whiteboard because I can't. I'm sorry, can you guys hear me? I tend to be having some technical issues with my camera, my network, and everything today. Anyway, please, let's go on. I'm just going to go ahead and use my whiteboard in the sense that the presentation, I don't know what's going on. So I said the DNA and RNA are what we regard as nucleic acid. So when you hear of nucleic acid, we are talking about DNA and RNA. There are other types of nucleic acid too, but the most common one, the two common ones that you have to know is the DNA and RNA. And the nucleic acid, when we talk about a nucleic acid, it's an aggregation of nucleotides. So there's one nucleotide that is joined to another nucleotide that is joined to another nucleotide. So a chain of nucleotide is what we call a nucleic acid. And nucleic acid is what we in layman term call a DNA and RNA. So DNA and RNA are a type of nucleic acid, while nucleic acid are a chain of nucleotide. Then a nucleotide is when we have a sugar, a ribose sugar from your basic organic chemistry, joined to what we know as a base. The base, we are going to talk about what a base is, and a phosphate group. So when we have sugar, a base, and a phosphate group, that is what we call a nucleotide. And when we have 2, 3, 4 nucleotides joining themselves together, that's what we regard as nucleic acid. An example of nucleic acid is DNA and RNA. Are you guys following me?
Yeah.
Yes.
So now, that will lead me to the question, what are the type of base, you know? When we talk about base, what are we referring to? Anyone?
Yeah, we have DNA 4 bases, RNA also 4 bases. DNA guanine, adenine, tyrosine and cytosine, RNA. RNA is adenine, uracil, thymine. See, it's the same 4 with DNA aside the T, but I need paper to look for it. Yeah.
Okay, yeah. Basically, when we talk about a base, you guys are getting it. You just have a little bit of misconception, but that is normal. Of course, that is what we regard as base. The component, like I mentioned from earlier, I told you guys that nucleotide are the building block of a DNA. When we are talking about DNA, the DNA does not just contain, how do you say, the DNA contain adenine, right? What's the difference between adenine and adenosine? Does anyone know the difference? When we say adenosine and when we say adenine, what is the difference between the two? Anyone? You don't know. Okay, and that is where the misconception lies. Adenosine is a base. Adenine, this is a nucleotide. They are two different things. So a base, like I told you guys, they are nitrogenous compound, either as a single branch chain or double branch chain that have nitrogen in them. And that's why they are called nitrogenous base. They contain nitrogen. This is a base. Do you understand? Then to this base, when you attach a sugar, that is ribose sugar, and you attach a phosphate to the base, that's when the name changes from adenosine to adenine. Do you understand? So this structure, this adenine, is what is found in the DNA, not adenosine. Adenosine is the base, of course, it's part of it, but it is attached to the sugar and to the phosphates. And together, it's called a nucleotide. Together, these three structures are called adenine. But when you remove the only base out of it, it's regarded as adenosine. Do you guys understand?
So
So
sorry.
yes.
You said adenine is a nucleotide.
Yes.
I'm kind of browsing right now, and they said
I'm
adenine is
confusing.
the purine base,
I'm
and
confusing.
adenosine is the nucleotide molecule.
Thank you. I guess I'm confusing the names for each other. Okay, thank you for that. So this is the base. Adenosine is the base, right? Right?
Adenine is the base. Adenine is the base. Adenosine is the nucleotide made up of adenine, phosphate, and
Thank you. Adenine is the base, and it's just the same thing I've been saying, just that I'm confusing the name together. So adenine is the base, while adenosine is the nucleotide. So adenosine is what is found in DNA. The nucleotide is what is found in DNA, not the base. The base is part of the nucleotide. When the base is joined by sugar and phosphate, it's called a nucleotide. Do you guys understand? So it's like that for every other one of them. It's the same for every other one. Guanosine, what's the other ones called? Anyone, the other type. So it's all the same. They have what we regard as a base, and when you attach a phosphate or a sugar to them, they become a nucleotide. Now, I asked for the difference between a nucleotide and a nucleoside. right? Who can tell me the difference between a nucleotide and a nucleoside? Can anyone tell me the difference?
Nucleoside is nucleotide without phosphate group.
Thank you so much. So a nucleoside is the base. Like I said, the nitrogenous base. They can be single, distinct, or double, depending how we come across that now. Plus the ribose sugar, plus ribose. So these two together is known as a nucleoside. When you pick a nucleoside and you add a phosphate to it, it becomes a nucleotide. So this is a nucleoside plus phosphate. So now I'll go back to the base and start everything again so you guys get a bigger picture of it. So this is it. You start with the nitrogenous base. You start with the nitrogenous base, right? This is a base. Then you pick this base, you add a sugar to it, the ribose sugar to it. When you add the ribose sugar to it, it becomes what? It becomes what?
Nucleoside.
It becomes a nucleoside. When you pick this nucleoside and you add phosphates to it, it becomes what? It becomes a nucleotide. Are you guys getting it? When you pick nucleotide, then add another nucleotide to it, it becomes what?
Nucleic acid.
Thank you. It becomes nucleic acid. An example of nucleic acid is
DNA, RNA.
DNA and RNA. Basically, that's just the simple. So they are building blocks, one step at a time. The first step is the base. Then you pick the base, you add sugar to it. Instead of calling it base, due to the addition of sugar, it's now nucleoside. You pick the nucleoside, you add phosphate to it, it becomes a nucleotide. Then you pick 2, 3, 4 nucleotides together, they become a nucleic acid. An example of nucleic acid is DNA or RNA. Do you guys understand it now? Do you understand it?
Yeah.
Okay.
Yes.
Thank you. So now, when you now go back, we now go back to the beginning again about a base. There are 2 types of base. They're either purine or pyrimidine. So those are the 2 types of base we have, the purines or the pyrimidines. As purine has 2 members, which is what? The 2 members of purine are, what are the 2 members of purine?
Adenine and thymine.
Adenine and, is it adenine or adenosine? Which one is the base again?
Adenine.
Adenine and what?
Thymine.
And thymine. Any other opinion? The 2 purines are adenine and guanosine, or guanine rather, not guanosine. Adenine and guanine. Pure, and that's why some of you might have heard of the mnemonics pure as gold. Right? Pure for purine, as for adenine, gold for guanine. So that is a simple mnemonics for you to remember your purines. It's very important that you are able to differentiate purines from pyrimidines, and I'm going to tell you guys why. So these are the 2 purines. The adenine and the guanine are the 2 purines. Then we have how many pyrimidines?
2.
Anyone? 2?
Think
Any
3.
other person?
Oh, 3 then, uracil, thymine, and cytosine.
So cytosine, uracil, and thymidine. So I use the mnemonic CUT to remember the pyrimidine, CUT for CUT. That is how I remember my pyrimidines. Nobody, you really don't know the structure. You really don't need to know the structure. But the one thing you have to know that is very important is that purines have 2 rings, while pyrimidines have only 1 single ring. Do you guys understand? So the base, these are nitrogenous. We call them nitrogenous base because they contain nitrogen from your basic organic chemistry. You guys are aware of the amine group. The amine are the NH, the compounds that contain NH2 group. So they are called nitrogenous base because they contain this nitrogen, this amine functional group. So there are 2 types of purine, adenine and guanine, and there are 2 types or 3 types of pyrimidines, cytosine, uracil, and thymidine. And they are what the sugar and the phosphate is added to make what is known as a nucleotide. Then when you have 2, 3 nucleotides coming together, that is how DNA is formed. So that's why I said don't get it confused. It's easy to get it confused that adenine is a base, while adenosine is a nucleotide. Now I'm going to draw how a nucleotide, so I'm going to draw how a DNA looks like after you add them together. So all these are used in making DNA and RNA. Of course, I don't think I have to point out the ones that are found in DNA and the ones that are found in RNA. You guys does anyone know of the law of complementarity? What does the law of complementarity state? Anyone?
No.
I don't know if there's a law, but I don't know, a purine nucleotide binds with the pyrimidine.
This is what is called the law of complementarity. That is, in the DNA, adenine or adenosine is attached to thymine or uracil. Oh, in the DNA, it is attached to thymine. In the RNA, it is attached to uracil, while guanine is attached to cytosine. This is what we regard as law of complementarity. For every A, there's a T, or in DNA, for every A in a DNA, there's a T, and for every A in RNA, there's a U. For every G, there's a C. Do you understand? So the number of A that you find in a DNA should be equal to the number of T you find. The number of G that you find in a DNA should be equal to the number of C. And the number of A you find in an RNA must be equal to the number of U, which basically means the DNA and RNA, the difference is this one. You never find thymidine in RNA, and you will never find uracil in DNA. This is what is regarded as a law of complementarity. So basically, what is happening in DNA is this. You have your A, that is your adenine, that is attached to phosphates. You have your phosphate here, you have your sugar here. But this A is attached to T. The T also has its phosphates and has its sugar. Do you guys get it? Then this A is also attached, like I said, a DNA nucleotide attached together. Then when they come together, they also attach to each other sideways. So this phosphate is also attached to another phosphate, let's say a C. The C has its sugar, and the C goes with what? G. G also has its phosphate, has its sugar. So the phosphate is also attached to another, and this phosphate is also attached to the other. So that's what they go on, and they attach. So basically, that is what is going on in a DNA. But the most important thing about this is, first, you have to identify, you have to know that purines have 2-ring chain, while pyrimidines have a single ring chain. Number one, you have to know the element, the types of purine, the 2 members of the purine group, which is adenine and guanine, and you have to know the three members of the pyrimidine group, which is cytosine, uracil, and thymidine. And you must be able to know their metabolic pathway, which is going to take me to the next slide. Any question before I move on from that? Any question before I move on from that?
No.
Okay. Okay, so I'm going to share my screen now. Basically, that's just like what I've said. A nucleic acid are assemblies of nucleotide, which consists of three components: a nitrogenous base, a carbon sugar, and a phosphate. A nucleotide, is an RNA or DNA monomer. They can be used as energy. ATP is also a type of nucleotide. Like I said, there are other types of nucleotide. Physiological, they can be used as physiological mediators like cyclic AMP, cyclic GMP. I'm going to talk about those later. And the sources of nucleotide, of course, this is very important. You have to understand that you can either get your nucleotide directly from your food, or you can synthesize the nucleotide in your body. And the synthesis of nucleotide is through two pathways, either as de novo synthesis, that is, you synthesize it new, or you salvage the ones you have in your body, you avoid breaking it down. Do you guys understand? I'm going to go over it. So, like I said, these are base. The purines have two members, guanine and adenine, and they have two rings. And the three members of pyrimidine are cytosine, uracil, and thymine. They have a single ring. The sugar, this is the sugar. There's nothing very important you have to know about the sugar. Just so, like, the one difference also among the difference between RNA and DNA, like I mentioned earlier, the presence of thymidine in one and the presence of uracil in one. The second difference is in the pentose sugar, in which, and that's why it's called ribose and deoxyribose. DNA is deoxyribose, right? And RNA is ribose. The difference is also in the sugar, in the sense that the two position, the hydroxyl group in the number 2 position in DNA is absent compared to that of RNA. So that's the second difference between DNA and RNA. I've explained the difference between nucleoside and nucleotide. A nucleoside is basically a nucleotide without phosphate, or a nucleotide is a nucleoside plus phosphate. So like you guys can see from the diagram, here we have an example of nucleoside. As you can see, we have the nitrogenous base, we have the sugar in the nucleoside. But in the nucleotide, we have a phosphate that is attached to the nucleoside. So we are going to move on to the purines and pyrimidine synthesis. The synthesis of purine and pyrimidine, do you actually need to memorize the pathway? No, you don't need to memorize the pathway. The one thing that you have to know in this pathway that is very, very important is actually the pharmacology of this pathway. The pharmacology of this pathway is very, very important. Right? Who can tell me, in a cancer patient, what is happening in cancer? What causes cancer?
Violation of cell division.
Violation of cell division.
Yeah.
Exactly. When you mean in medicine, when you are defining something, it's very good to be specific. When you mean violation of cell division, somebody like me might interpret it as reduction in cell production. Another person might interpret it as increase in cell production. Do you understand? Another person might interpret it as change in the cell type. So when you mean violation, you have to be specific. So what type of violation is occurring exactly in cancer?
Increased cell division.
Increase in cell division. That is one type, although that's the most important type, and that's what we are concerned about. But also the change in cell type also does occur in cancer. But okay, there's increase in cell number. And the whole idea of how does, that's for whoever is teaching you guys cell biology, that's the person's job. But anyway, how does the cell divide? what is the main component that determines the division of a cell?
Genetic material.
The what?
Genetic material.
Yeah, every cell has genetic material. What is happening exactly when we say a cell divides?
Proteins.
You said what?
Proteins, like they make new proteins.
Yeah, they make new proteins. How do they make new proteins? What's the first step?
Translation and transcription.
Okay, let me go ahead. What's the first step in cell division? What happens first when a cell divides? What?
Replication of genetic material.
The replication of genetic material, exactly. And that genetic material is what? What's the specific genetic material?
Chromosome.
DNA.
Exactly. Thank you. DNA. That is the major thing that happens. Your cell, you have your cell that contains the nucleus that has the DNA, right? This cell cannot divide except the first step in DNA. If you guys have done cell biology, you must have talked about the cell cycle. The cell cycle has 2 stages or 3 stages, right? The S phase, and the M phase, right? What happens during the S phase of cell division? What happened? What happened? What happened in the S phase of cell division?
DNA synthesis.
Exactly, DNA synthesis, not just DNA synthesis, DNA replication. So this DNA has to double itself, and that's what is called a chromatin. That's the difference. I know some people, if I ask the difference between chromosome and chromatin, a lot of people might not know the difference. A chromosome is just basically a double strand of DNA, right? This is a chromosome. A chromatin is when 2 double strands of DNA, that is 2 chromosomes joins together, is called a chromatin. But as a whole, we still count them as one. Do you understand? So the first thing that happened is duplication of this chromosome. This duplication, that is duplication of DNA. And I'm telling you guys that to make DNA, you need what? In order to make DNA, you need what?
Nucleotides.
Exactly. To make DNA, you need nucleotide. Nucleotides are the building blocks for your DNA, which means if you have nucleotide in place, DNA replicates itself, right? Divide, makes more cell. So if there's anything you want to do to stop this cell division, what is the first thing you have to do? What's the first thing you have to do?
Stop DNA synthesis and replication.
Exactly. So once you stop the DNA synthesis and replication, the cell will not be able to divide. And in order to stop the DNA synthesis and replication, what you need to do first and foremost is what? Make the nucleotide unavailable. Since they need the nucleotide, it's just basically you have the blocks to build a house. If you want to stop someone from building the house, the first thing you do is just take hammer and something and just go to wherever the person lays block down and go and break all the blocks. Once you break all the blocks, there's no way that person can go ahead and build the house. So that is why I said the most important thing about that pathway is in pharmacology, that is in the treatment of cancer. If you can find any way whatsoever to stop the synthesis or the availability, anything that will reduce the availability of nucleotide, will lead to reduction in DNA synthesis or DNA replication, and anything that leads to that leads to decrease in DNA synthesis will lead to decrease in cell replication. So that is why it is very important, and that's why that particular pathway is important in the cancer treatment. Another way that pathway is also important is in anemia. It's just the same. It's just the same ideology. Anemia too. You are trying to make up new red blood cell. If there is no DNA present, there is no new red blood cell. And when there's no new red blood cell, your patient has anemia. So that is for krok, that is the most important thing to know about this pathway. And that's why you see these drugs that are present. These drugs inhibit a particular enzyme in this particular pathway. Those drugs inhibit particular enzymes in this particular pathway. So the most important thing you have to know is the enzyme that is inhibited in this pathway and the drug that inhibits it. So you have to know that hydroxyurea inhibits ribonucleotide reductase, 5-fluorouracil inhibits thymidylate synthase, and we have methotrexate, timetropine, and pyrimethamine that inhibits the hydrofolate reductase. Do you guys understand?
Yeah.
So it's very, very important. Very, very important. Another thing that is important in this pathway is this whole cycle of folate. Folate, which vitamin, what is another name for folate?
Vitamin B.
E9.
Vitamin B9. Thank you so much. And it's also the same ideology, like I told you. This particular vitamin is very important. This folate, the hydrofolate. Folate is basically converted to dihydrofolate. The hydrofolate is then converted to tetrahydrofolate. Tetrahydrofolate is converted by adding a methyl group that is a CH addition to it in order for it to donate that CH to something else. And I'm going to show you guys what I'm talking about. I still don't like this screen sharing anymore. It's the same thing. So imagine in the DNA synthesis, right? You have the folate. When you bring your folate is converted to dihydrofolate. But first of all, I'm going to try, I don't know the exact structure, but I'm going to try to draw it. The difference between uracil, let's say, I told you guys they have one chain. Let's say this is uracil. This is what? Let's say this is uracil. I know the structure might be wrong. You don't need to memorize the structure. But when you add this CH3 to uracil, what do you get?
Methyl uracil.
Methyl uracil. There's another name for methyl uracil. I like it. You must know organic chemistry very well. This is methyl uracil, but there's another name for methyl uracil. Anyone? Can you guys see my screen?
Yeah.
The other name for this methyl uracil is thymidine. And I said uracil is found in what?
RNA.
But you have thymidine in what?
DNA.
So that's basically the difference, right? And that's why some people say RNA comes first before a DNA, in the sense that the RNA has uracil. It's from the uracil that you make your thymidine, because every other one is the same. So you have your adenine, you have your guanine, you have your cytosine that are the same for the three. The only that are the same, the three are the same for the two. The only difference is in the uracil and the thymidine. But you have your uracil, you need to make your thymidine, right? And to make thymidine from uracil, and that's the whole essence of that de novo pathway of synthesizing pyrimidines. That's the whole idea, to make thymidine, because we need to make DNA, but we have uracil. And in order to make thymidine from uracil, you have to add this CH3 to your uracil. Do you guys understand? So in order to add this CH3 to your uracil, which means this CH3, that is the methyl group, must come from somewhere, right? Now, where does the methyl group come from? Anyone? Anyone?
From the dihydrofolate.
Thank you so much. So the dihydrofolate, so you have your folate, that's converted to dihydrofolate. The dihydrofolate is then converted to tetrahydrofolate. Then this tetrahydrofolate is the one that is able to carry this CH3. Do you get it? That's where this CH3 comes from. I'm going to get to that later on because they are all connected. The reason why the deficiency of folate causes anemia is just this whole pathway in such a way that if you don't have folate, right, you can't make tetrahydrofolate. Tetrahydrofolate cannot take this CH3. Then this CH3 cannot be donated to uracil to make thymidine. And if you don't make thymidine, you don't make what?
RNA. I mean DNA, sorry.
You don't make thymidine, you don't make what?
DNA.
DNA. And if you don't make DNA, you don't make what?
Cells, basically.
You don't make new cells. So that's the basic idea behind everything that happens, either in treatment of cancer or either in anemia. Anemia basically is you don't have enough red blood cells, right? So basically that's the whole idea. And why folate is important, but not only folate is important, this CA3 is also very, very important. Where this CA3 comes from, does anybody have an idea where this CA3 comes from? I want to see the gurus. Does anyone know where this CA3 comes from? What? It comes from? Come again, please. I didn't hear you. Yes. Tetrahydrofolate is just like a carrier, just to carry it and give it to uracil so that it can make thymidine. But there's somewhere that the tetrahydrofolate also collects this CH3 from.
Folate.
From what? Anyone? Folate, no. Folate is just basically, it's just that folate cannot hold the CH3 by itself. So it is converted to dihydrofolate. Dihydrofolate is just basically folate, but there is a difference in structure. A double bond is removed. So folate, dihydrofolate, and tetrahydrofolate, they are the same thing. It's just basically change in structure. You guys have heard of isomerism. They are basically isomers. They are the same thing. It's just a change in structural. They are just structural isomers. Folate, dihydrofolate, tetrahydrofolate. Is because they have to change that form to be able to accommodate that CA3 at the end. But that CA3 has its own source. There's somewhere else it comes from. And over time, when I get to it, I'm going to make mention of where it comes from. But for today, it is not part of our topic. But you guys can try to find out and, surprise me in the next class. So basically, that's just the whole idea, which means not only are these enzymes very important, also the name of the drugs that inhibit it, you have to know it. And inhibition in any step in this thing will lead to what? Reduction in DNA synthesis. So as you guys can see, you have your CO2 and glutamine, blah, 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 that comes together, then with carbomoyl phosphate, form carbomoyl phosphate, blah, 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 then come to uridine diphosphate. You see, your uridine diphosphate, as you can see from here, is what is converted to thymidine by thymidylate synthase. Can you guys see that bottom corner where we have so the base, that's the whole idea. What's going on with this thing? Okay. So as you can see, you guys, UDP basically means uracil diphosphate. The uracil diphosphate converted to deoxyuracil monophosphate is what is converted to thymine monophosphate. And the thymine monophosphate is done just by adding that CH3 group, that methyl group you see. By adding that methyl group to it, then you make your DTMP. And that particular enzyme that catalyzes that particular place is thymidylate synthase, and it can be inhibited by 5-fluorouracil. But the most important step you have to know is that the conversion of dihydrofolate to tetrahydrofolate is catalyzed by dihydrofolate reductase, just by the name, dihydrofolate reductase. How to know the name of enzymes I'm going to teach you guys soon, so don't worry about that. But basically, the hydrofolate reductase convert the hydrofolate into tetrahydrofolate, right? And the drugs that you must know that is very important is methotrexate. Trimethoprim and pyrimethamine are antibiotics or antiprotozoan. So we use them in microbiology. But the whole idea is the same idea. If you want to kill a cell in microbiology, what causes disease is the replication of bacteria. If you're infected by one bacteria, you are not going to have any sickness. But when the bacteria multiplies in your body, right, is when it causes symptoms. So the one way you can stop yourself from displaying symptoms is stop the replication of the bacteria. And for a cell to replicate, DNA has to be replicated. For DNA to be replicated, bases have to be synthesized. That is either purine or pyrimidines have to be synthesized, especially pyrimidines have to be synthesized for DNA to be replicated. So if you inhibit the synthesis of nucleotide, that is the synthesis of purines or pyrimidine, you directly inhibit the synthesis of DNA and indirectly inhibit cell replication. Do you guys understand? There's also one basic, very important biochemical point that you have to know in this pathway is UMP synthase. You see, from that carboxyl phosphate to orotic acid, then from orotic acid, you add PRPP. PRPP is basically the sugar to make UMP, uridine monophosphate. And the reaction is catalyzed by UMP synthase, right? Who can tell me when this UMP synthase is deficient, what do we call the name of the pathology? Deficiency of UMP synthase causes what? Anyone?
Orotic aciduria.
Thank you. Basically, naming disease is very, very simple, especially when it comes to biochemistry. If I tell you that an enzyme is deficient, just look up which particular product comes before the enzyme. That is the name of the disease. Whichever enzyme, anywhere I get to, and I tell you there's a deficiency in one enzyme, look at the product that is at the top of that enzyme. That product is going to be accumulated on the body, and that is the name of the disease, orotic aciduria. That is, it will increase in the urine. Orotic acid, due to the excess in the body, is going to spill into the urine. Of course, if you don't have UMP synthase, your orotic acid cannot go forward into UMP, which means there will be backup of whatever is behind it. Uric acid will be backed up, carboxyl phosphate will be backed up. Do you understand? CO2, glutamine, blah, blah, will be backed up. So the disease is uric aciduria. So the deficiency of UMP synthase causes uric aciduria. And the basic thing is that you find excess uric acid in the blood that spills into the urine. So when you test the patient's blood when you test the patient's urine, you see uric acid. So basically, I've told you guys, no, very important, this enzyme, the enzyme that catalyzes UDP to deoxy-UDP. What's the difference between UDP and deoxy-UDP? Anyone? UDP and deoxy-UDP. What's the difference? Anyone? Can anyone tell me the difference? While I'm still drawing.
Probably as the name suggests, you said UDP and deoxy, so probably in the UDP there's a presence of an hydroxyl group, more likely, and then in the deoxy, probably probably hydroxyl group is missing. I'm not too sure.
Okay. Yeah, basically, this is the difference. This is UDP. And this is deoxy-UDP. What's the difference?
The absence of an hydroxyl group.
the absence of this hydroxyl group is what changes it from, like I said, there are two differences between RNA and DNA. The first is what?
The same thing, absence of the hydroxyl group.
The first
So,
is the absence of thymidine in RNA and the presence of uracil. This is the first difference. The second difference is the absence of OH in the second carbon position. This second carbon. This difference, that's the difference between UDP and deoxy-UDP. So when you remove this OH here, it becomes deoxy-UDP. And which enzyme is responsible for that? Which enzyme is responsible for that?
UDP synthetase.
Okay. The enzyme is what? Any other opinion? Any other opinion? The enzyme that is responsible. Just think, what happened here? What happened here to here? In layman terms, if you want to explain what happened, anyone? The best way to remember it is, this is RNA, right? This is RNA. I told you guys, this is an RNA. So RNA is what? Ribonucleotide. The best way to remember is it is reduced. That is, this OH is reduced from it to give us this. So in order to know the name of the enzyme, just think of it, ribonucleotide reductase, the enzyme that reduces ribonucleotide. Do you guys understand? Is anyone still with me? Do you guys understand, or I should explain again?
Yes, understand.
So it's very, very simple. See, biochemistry is, how do I say, it's just a language. You just have to speak the language. And to speak the language, it is very, very easy. You have something, it has OH, it has two OH. Then I have someone that reduces it. It reduces the number of OH and turn it into one. And in order, because there are so many other reductions in other places, so that I will know where the reduction is happening, I pick the name and remember that, oh, this particular person reduces RNA. So RNA reductase, ribonucleotide reductase, simple as ABC. So that is the enzyme that converts UDP into deoxy-UDP to make the DNA component of it. So, and that enzyme is very important in such a way that if the enzyme is deficient or is inhibited, then you have RNA, but you don't have DNA, because the deoxy part of the DNA cannot be synthesized. Are you guys still with me?
Yes.
So basically, that is it. So that's basically the de novo synthesis. The catabolism. What's the difference between anabolism and catabolism? Anyone? Does anyone?
catabolism is breakdown, anabolism is basically build up.
Thank you so much. Anabolism is basically build up, we are making, and catabolism is we are breaking down. The catabolism of pyrimidine is not very important in such a way that it is completely broken down and or recycled by enzyme. And this is where I said it's important to know it's basically broken down to ammonia. And or if it's not totally broken down to ammonia, it is recycled back to make another, nucleotide. Using the pyrimidine, we make another nucleotide, then make another nucleotide, then make DNA. So it's no problem. Then for purine, basically in the metabolism of purine, the one thing you have to know, the one thing that's very, very important in the metabolism of purine or in the synthesis of purine that you have to know for your exam, actually nothing. In the synthesis of purine, you actually need to know nothing. Just read through this particular slide, but there's nothing very, very specific that you have to know in the synthesis of purine. The most important part is in the catabolism of purine when you are trying to break the purine down or when you are trying to salvage it. So like I said, when you make your purine or when you make your nucleotide, right? You make your DNA, then you make your cell, then the cell dies. It does what? It spills the DNA. Then the DNA is broken down to these component nucleotides. The component nucleotides then are broken down into the component nitrogenous base, right? Then you have your nitrogenous base floating around in the blood. And the nitrogenous bases are what? Your nitrogenous bases are your what? Your purines and your pyrimidines. For pyrimidines, it has two fates. The fate is it's broken down to ammonium, or it's salvaged back to make nucleotide. You understand? It's salvaged back to make nucleotide. The same thing with purines too. Purine is broken down to uric acid. See the difference? Broken down to ammonia. This is broken down to uric acid, or it's also salvaged back to nucleotide. So your nitrogenous base, after they are used to synthesize DNA, then the DNA breaks down, they start floating around in the blood. They have two fates. It's either they are broken down, pyrimidine is broken down to ammonium, or purine is broken down to uric acid, or they are both salvaged. That is, they are both saved from being broken down into uric acid or into ammonium. So basically, in the breaking down parts, blah, 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 the enzymes involved, blah, blah, you don't need to memorize that. The one important enzyme you need to memorize is in the salvage pathway. That is, when you break down your whatever and you produce inosine or guanosine, they are converted to hypoxanthine or guanine. This hypoxanthine and guanine can be salvaged back to make guanine monophosphate, which is a what? Guanine monophosphate. Guanine is a what? Guanine is a what? Anyone? What class is guanine? Exactly. Guanine is a base. Thank you. And guanine monophosphate is a what? Guanine monophosphate is?
Nucleotide.
Thank you. Is a nucleotide, which means you can salvage your guanine back to guanine monophosphate, which is a nucleotide, or you can salvage your hypoxanthine back to inosine monophosphate, which is also a nucleotide, right? And the enzyme that is responsible for that salvage is what we know as IGPRPP, hypoxanthine guanine pyro, whatever. I can't even remember. Even I can't pronounce the name of the enzyme. I never tried to. I just remember it as the HGPRT. That is the enzyme. And the reason why it is important is because the deficiency of that particular enzyme, your inability to salvage this particular pathway, your inability to convert hypoxanthine and guanine into inosine monophosphate or guanine monophosphate leads to a syndrome that is known as Leishman syndrome. Just let me give it to you straight, simple as ABC. When you get into the exam hall, don't think too much. Whenever they just mention a question that talks about self-mutilation, since I have been in medical school, that is the one pathology I've come across that has to do with self-mutilation. And when we say self-mutilation, what's self-mutilation? Anyone? It's just English. What's
Harming
self-mutilation?
yourself.
Exactly. Harming yourself. The one pathology that have that symptom is Leishman syndrome. And Leishman syndrome is caused by the deficiency of hypoxanthine guanine PRT. I don't know the meaning of the PRT. Not that I don't know. I actually forgot. I actually don't care. I don't personally. That's what I tell you guys. You don't need to memorize the name of the enzyme. You just have to understand, especially the steps that involve the tying of it to pharmacology or tying of it to pathology. Because we are in medicine. That is the most important aspect of it. You guys understand? Or, like I said, if they are not served back, they are broken down into uric acid. And this is why it is important. Pyrimidines are broken down into ammonium. Ammonium can be excreted by the liver into the urine, simple as ABC. Excess ammonium, I don't know, encephalopathy, that's when the liver is problematic. It causes nothing. But excess uric acid causes what pathology? Anyone? Excess uric acid. What does excess uric acid cause?
Gallstones.
Gout, gallstone.
Yeah,
You
gout.
are all correct. Exactly. Excess uric acid leads to what is known as gout when it's accumulates in the joint. And when it's excess too, it leads to kidney stone when it's excess due to the excretion from the kidney. And this is where you have to know the difference between purines and pyrimidines. And that's why purines can cause disease. The breakdown of purine, when it is in excess, can cause you a disease. But the excess of pyrimidine does not cause anything. Do you guys understand? So the essence of purine breakdown causes gout, but the essence of pyrimidine breakdown, you are fine. Because your ammonium deliver will handle it, but the uric acid will accumulate in your joint, causing gout. So, and that leads us, gout is basically, joint pain. But the most common thing that you guys will come description is the big toe, inflammation of the big toe, or whatever description you guys see. It's talking about gout. And in that also, I will go back and give you guys another very, very important tie-in to pharmacology, in the sense that when a patient has gout, one of the drugs that is used in treatment of gout is allopurinol. Allopurinol inhibits the enzyme that converts xanthine to uric acid, and the enzyme is known as xanthine oxidase. Like I told you, most enzymes will always have a name of the precursor and the action that it does. That is what they act on, the name of what they act on, and what they do to it to form a new product. Do you guys understand? So xanthine is the, what do we call it? There's a term we use in chemistry. There's a term we use in chemistry to talk about the other part of the, the equation. Substrates, thank you. Substrate. Xanthine is the substrate. Then xanthine oxidase is the one that will take the substrate, do some process on the substrate to give you the product. The product is uric acid. Do you understand? It's basically xanthine is your uncooked rice. Your xanthine oxidase is your fire, and your uric acid is your well done rice. Do you understand? So it's the fire that will cook the xanthine to give you the uric acid. So basically, if you don't want to have too much cooked rice, you can just, cut the fire. Then if you don't want your rice to be overcooked, you can just off the fire once you are at a acceptable level of softness of your rice. Do you get it? So one way to off the fire is allopurinol. Allopurinol will off your fire for you. And the fire in this case being xanthine oxidase. So it's very important in order to treat uric gout, which is accumulation of uric acid, you just have to make sure that there is a reduction in production of uric acid. And one way to reduce the production of uric acid is actually by inhibiting the enzyme that leads to its production, and the enzyme is xanthine oxidase. So you guys should keep note of that. Like I told you guys, this is the structure of a DNA. You have your nitrogenous base, that's the T, that is attached to another nitrogenous base, that is the A. You have your sugar that is attached to your phosphate. Do you understand? Then you have the other O of the sugar that is attached to the phosphate of another base. So this bond, that's why we say a nucleic acid, they said, is a polymer that are joined by the 3 and 5 end. The 3 end is the end of the sugar. The O end of the sugar is what we got as 3 end. The 5 end is the phosphate part. And that is where on the linear, that first line, the line on a linear portion, that is how they join to each other. The phosphate group bind to the OH group through what we know as the phosphodiester bond. That is what linked them together. But then also we have a disulfide bond that links the T and the A together. So in a single strand of DNA that is in the linearly, the nucleotide is joined to another nucleotide through the phosphodiester bond. But from one line to the other line, the force between them is what we know as a disulfide bond. So basically, this is another organization of DNA. And yeah, nucleosome, nucleotide, and chromatin, basically I've made a point about them. This is a DNA, right? The double-strand DNA. Then the DNA is wrapped around some tires. You understand? Those tires are called the stones. So when it's wrapped around them, that's what is known as chromatin. And when the chromatin can be expressed, that is E-chromatin, or they can be packed together, that is heterochromatin. So that's basically it. And you have to know your type of histones. It's just basically numbers. Histones 1, 2, 2A and 2B, then 3 and 4. Those are the type of histones. I'm going to give you a diagram. I'm going to try to give a schematic. Have you guys not been seeing my screen?
You were sharing your screen.
Okay. So basically, what is the whole histones, non-histones story is about? So it is a DNA. You have 2 lines. Then let's say someone placed tires like this. You have 2 lines that are like 10 meters long. You understand? But come on, 10 meters cannot fit into your body. So what you do is there are some tires. These tires are histones. These are histones. H2A, H2B, H3, and 4. They are like in 2 pairs, you understand? So these are like tires that are around, but this is 10 meters. These tires practically, with the distance between them is just like, let's say 2 meters. This whole length is like 2 meters. Then you want to wrap around this thread of 10 meters. What do you do? Just pick the wrap around, wrap around, wrap around, wrap around, wrap around, wrap around, and wrap around. Basically, this is it. The histones with the DNA wrap around it, that's a chromatin. Basically. So instead of the 2 meters long thread, you now compress it into 2 meters. Do you understand? Then you have your another histones, that's the H1, that comes outside. You know, the DNA is wrapped around H2A, H2B, 3 and 4. Then you have H1 that will come around and strengthen these ones again, making the 2 meters into, let's say, 1 meter. You understand? Join them together again. So it's outside. It's not together with the other ones. Then together, they form a chromatin. Now, this chromatin can be so packed together, like the tires can be so packed together that there is no space. This is what we know as heterochromatin. And in a cell structure, it appears dark. whoever teach you guys cell biology should teach you that it will appear dark. And because they are so packed together, the DNA is so packed, you understand? So if you want to see a portion of the wire that is wrapped around there, you can't see one particular portion. And that's why this particular portion is not transcribed. It cannot be transcribed because that particular portion cannot be read by your enzymes. And this is also what happens in bad bodies in women. The presence of bad body is just a very condensed DNA around histones that is so dark, it's so compressed that the DNA polymerase cannot read it to transcribe. Or the wires, the tires and the wires can be, of course, they are packed together, but not so packed together. You understand? Just a little bit packed together so that the DNA polymerase can read it. And this is what is known as heterochromatin. This particular portion can be transcribed by the DNA polymerase. So that's just the difference. And that is how the difference in human being is made. That is, some portion of your own DNA are so condensed together, they are in heterochromatin form. So it is not transcribed in you, while that particular portion might be like a little bit loose, that is e-chromatin in my own body, and they are transcribed in my body. Do you understand? Any question before I conclude the molecular biology section? Any question? Please ask your question. You guys don't have any question? Okay, so thank you. So which means I'm going to move now. Now we've made our DNA, right? From the whole talk, from our base, add sugar to it, add the phosphate to it, then join them together, we've made our DNA, right? Simple. Now we have one DNA, or we have DNA, and we've also talked about the chromatin, the chromosome. So now we have a cell, one single cell. My drawing of the cell is terrible. Let's say now we have a cell, nucleus that contains the DNA, and we have the cell, right? Now we want to make new cell. How do you make new cells? How do you make new cell? Don't forget that this cell, this is the nucleus. It has the mitochondria. It has the Golgi apparatus. It has the endoplasmic reticulum, the smooth and rough, right? Now we have one single cell, and that's just how it happens. Let's say fertilization. No, before even fertilization, even when fertilization occurs, you have one egg, which is a single cell, then that is fertilized by the sperm. So you have one cell now, one single cell. What happens to make a human being? What happens? Anyone?
Cell division.
We have cell division will occur, right? Exactly. And a cell is made up of organelles. Those organelles are proteins. Let me just put it down now. They are proteins, which means they are all proteins. The organelles are proteins. And you want to make a new cell, which means you have to make these new proteins, right? So that's the whole ideology about cell division. You want to make proteins. The proteins we are referring here to are organelles. You want to make new mitochondria. You want to make new lysosomes. You want to make new endoplasmic reticulums, right? So, and the genetic material that you are going to use to make them is called what? The genetic material you are going to use to make them is called what? How do you know what, how does the body know what to make? What tells the body? Where does the body get the blueprint exactly to build new cells? What is the blueprint for the new cells?
Is the DNA now?
Exactly. The DNA is the new print, is the blueprint. It's who is teaching you guys genetics? Have you guys started genetics. Davis, are you on this call? Davis. Seriously, I need to know who is teaching you guys genetics anyway.
Yeah, we
So-
haven't started that yet.
You haven't, I told you, I don't know how you guys did the timetable anyway. So basically, the blueprint is DNA, right? That we use to make new cells. So the first thing in order to make a new cell is to duplicate this new DNA, because for the new cell to have its own nucleus, that's to tell you that the nucleus is the first one to form. So once you have the nucleus and the DNA, you understand? Once the new cell has its own nucleus and its DNA, it can go ahead to make its new proteins, the endoplasmic reticulum, the cytoplasm, using this DNA. So the first step is to make the DNA of the new cell from the DNA of the old cell. And the process of making the new DNA of the new cell from that of the old cell is called what? The process, I'm going to come again. Now, we have one single cell. You want to make a new cell, right? And in order to make the new cell, you need a blueprint. In order to make the blueprint, which is the DNA, once you have the DNA, the blueprint for the new cell, that is all what the new cell needs. From its own blueprint, it can build itself, right? So in order to make that blueprint, you just want to photocopy the blueprint of the old one, which is the DNA. Making a new copy, that is making the photocopy of the previous DNA to the new copy, is what is known as what?
Replication.
Replication.
Thank you. That is what is known as replication. That is the first step. You have to copy this particular blueprint. You have to take it to that shop behind the hotel, Hakim Hotel. You have to take this copy, that is the DNA, you have to take it to her so that she can photocopy it. Her photocopying it. Photocopy is what is known as replication. Basically, replication is photocopying. So you are photocopying the DNA of the existing cell to make another copy for the new cells you want to make. So that is replication. DNA to DNA, exactly. It's photocopy, nothing changes. It's the same thing. And this is replication. That is the first step in making a new cell. And the whole ideology of replication is knowing the enzymes that are involved, right? So I told you guys that when the DNA is too wrapped up, right? That when the DNA is too wrapped up, it cannot be transcribed, or it cannot even be read by the DNA polymerase. So the first thing, don't forget that they are joined together, This is a chromosome or a DNA, two strands of DNA joined together, right? So the first thing you need to do in replication is make sure you untangle some portion, like lose it, create space, lose it in the middle, so that the photocopy machine can photocopy. Do you understand? So that the photocopy machine I thought your class is 2 hours. So basically, I'm just going to, I thought the class was 2 hours, but I had this an hour, 30 minutes class. So basically, the process of making a DNA from a new DNA is known as replication. And the one thing you have to know about replication is that the enzyme that is responsible is called DNA polymerase. I told you guys, just know the name of substrate. The substrate here is DNA that you are making to use another DNA. Polymerase basically means photocopying. Wherever you know hear of polymerase is doing the photocopy. So the enzyme responsible DNA polymerase. Do you understand? That is the enzyme that is responsible for making of new DNA. There are other enzymes that are involved, like LEKase. LEKase is responsible for unwinding the tight chromosomes, the tight DNA together. So we have topoisomerase that are, when you when you untighten those DNA, there's a possibility for them to actually, it's just like when you leave a thread, when it's too long, there's a possibility of it getting entangled. When you leave your earpiece, right? When your earpiece, when you leave it, when you come back, you don't know how it happens, but it gets entangled anyhow. That you have to be sorting it out. The enzyme that avoid the DNA getting entangled is known as what is known as topoisomerase. So these are just basically enzymes that you have to know, but the most important is DNA polymerase. The making of RNA from DNA, you have DNA, right? The DNA is your template. And you want to make RNA, the process is called what? You've made the new DNA. From the DNA, you want to make RNA. This process is called
Transcription.
transcription. Thank you. And the enzyme responsible for transcription is called RNA polymerase. So the enzyme responsible for making DNA from DNA is DNA polymerase. The enzyme that is responsible for making RNA from DNA is called RNA polymerase. The process is called transcription. Then the process of making proteins, which are organelles that I'm talking about, is the process of making protein from RNA is called
Translation.
translation. Thank you. So basically, in conclusion, this is it. First of all, you make DNA from DNA, called replication. Then you make RNA from DNA. It's called transcription. Then you make protein from RNA. It's called translation. We have different types of RNA. We are not going to, but basically from mRNA, we are not going to dwell into that. So basically, that is the whole picture. And the enzyme involved here is DNA polymerase. The enzyme here is RNA polymerase. The enzymes that are involved here is what? There's no enzyme that is particularly involved, but there's structure that are involved that is called what? Ribosomes. The unit of ribosome, how it's done, how translation is done, and that we are going to talk about that in the next class. I thought it was a 2-hour class, but I'm informed it's an hour 30 minutes class. So I'm going to conclude the class here, and in the next class we are going to continue. I'm very sorry for my initial, I don't know, I was a little bit out of it at the beginning. I was having some little problem with myself, but anyway, I'm very sorry about that initial time wasting. I think that's one of the reasons why we couldn't complete what we set out for today. But that's that. Any question? You are free to ask any question you have about what has just been discussed.
No.
You said?
No question.
Okay. Any other person has any question? Or I'm free to go? Okay.
There's no question.
Okay. Thank you very much, Habib. So good evening, everyone. So I thank you once again for attending today's tutorials. I'm happy to see everyone here. So I would also like for you to inform your friends about the tutorial so we can have more people. That would be highly appreciated. Also, tomorrow is Sunday, and you guys are going to have a krok test of about 100 to 150 questions. So we're going to send a Google form link, and you have the whole day to do the test, but it's advisable to do it within 2 hours. So do it within 2 hours and submit your answers. But the test is going to be available for the whole day, so you can do it at any time you like, but please do it within 2 hours. So that's it. So if there are no questions, have a nice day. Good night, guys.
Good night.
Thank you very much. Good night.
Thank you.
Thank you. Bye-bye.